Picking up where we left off with still in meiosis 1. So we've talked prophase, metaphase, anaphase 1. Um, prophase, they are getting set up, centromeres are moving. Metaphase, and crossing over happens. Metaphase, they are lining up and orienting themselves. Anaphase, they are starting to separate. There are some things that can happen that we also need to know about. Non-disjunction is going to occur when problems with the mitotic spindles cause errors in the daughter cells. Non-disjunction means that it does not pull them apart in the correct way. This can occur if a tetrad of chromosomes don't separate properly during meiosis 1. It can also happen during meiosis 2. So anytime the chromosomes are connected by something, if they're not pulled apart effectively, I can get too many or too few chromosomes and stuff. Another thing that can happen is aneuploidy. If a gamete is fertilized, the resulting cell will not have the proper number of chromosomes. That is called aneuploidy, either too many or too few. Ploidy usually signifies that there's too many. Ploid or like plural. So prophase, metaphase, anaphase, and telophase 1, all part of meiosis 1. The chromosomes are still double-stranded, containing the two chromatids. So I have 23 chromosomes that still look like X's in one, 23 chromosomes that still look like X's in another. The double-stranded chromosomes migrate to the poles, and the number of chromosomes is technically considered to be haploid at this point. Telophase and cytokinesis is going to be the actual division of these two cells. So I now have two cells, each with 23 X-shaped chromosomes. Interkinesis, this is the time between meiosis 1 and meiosis 2. No DNA replication happens. So unlike during meiosis 1 where we have interphase to replicate my DNA, I don't have that, and for good reason, because I need to get half of the DNA. This is exactly like mitosis, but the DNA has already been replicated earlier, and it separates those double-stranded chromosomes or those X's into single-stranded or chromatids. So they will all end up with 23 single chromatids, which is half of the DNA that they need. They are haploids, 4 of them. Again, so interkinesis between meiosis 1 and meiosis 2. It's going to create haploid daughter cells because no further DNA replication happens. Prophase 2. Again, centrosomes move, and the chromosomes will start to compact again, move to where they're supposed to be, and the spindles will start to form as well. In prophase or prometaphase 1, during meiosis 1, versus 2, we see that there is not a lot of crossing over that's going to happen because on the top row, I've got my two chromosomes where crossing over can occur. In the bottom, I'm splitting two chromatids, and so crossing over doesn't really happen. So prophase 2, or we've got interkinesis, prophase 2, metaphase 2. The double-stranded chromosomes have lined up on the metaphase plate. The microtubules attach, and they start to pull each other apart. The actual separation occurs during anaphase 2. Centromeres, or the connection of those two chromatids, separates it, and they start pulling each other apart. Cytokinesis might already start at this point as well. Telophase 2 is when the nuclear membrane starts to form. The chromosomes can uncoil into their nucleus, and cytokinesis completes at this point as well. And so we have our 4 separate cells. So this is the process that's going to happen in humans. We call it gametogenesis, but it's basically meiosis in humans. Either spermatogenesis, 4 sperm cells are created, or oogenesis, which is the creation of eggs. What's interesting in this case is that during cytokinesis, there's an unequal division of the cytoplasm, which basically means that only 1 out of the 4 eggs survives. And think about the fact that this all happens when I, as a female, or other females, are in infancy, right? We get all of our eggs, and that's it. And so We call them polar bodies, but basically from one egg, only one new egg is going to be formed, and three polar bodies, or cells that eventually die and wither away, are created, which is why women have a finite number of eggs and males can continually produce sperm. So again, things we need to know, reviews. I would pause this slide, take a look. I'm not going to go into too much detail here. I think that you guys can look at this if you need to. Again, same here, pause it if you need to. We've kind of hit all the important parts of this. Why is this evolutionarily useful for us to know? Crossing over, random arrangement of chromosomes when they're on the metaphase plate, random fertilization once we do mate, and choice of mates can all yield a large number of variability in the population, which is a good thing. And so that is the end of all of our cell division material. We're going to move on over break, but you will have a test on this material as well as the other break material when you return, probably a Wednesday.